Okay, hi everyone. Good morning, or should I say afternoon or good evening, wherever you are in the world. Thank you for joining me today. My name is Monique Seymour. My background is in biotechnology and management, and I received my MSc from the University of Glasgow. I work for RapidNova as an international scientific sales executive. We are a startup CRO specializing in de novo protein sequencing. Our team has grown quite a bit since that picture was taken, so we'll definitely need to get a new one when the team reunites post-COVID. And if you have any questions, please type them below and we'll get to them at the end of the presentation. So over the past few years, RapidNova has successfully completed over 2,000 protein sequencing projects. So we've really accumulated expertise in protein sequencing and its application in providing research insurance. So to begin, what would you do if a tree fell on your house today? Or what would you do if someone rear-ended your car? What about tomorrow if you lost your favorite antibody? Chances are you probably have pretty direct answers to the first two questions. You'd give your insurance a ring and tell them to help you sort out the freak accident. But what is your answer to the last one? Getting insurance in our day-to-day life seems like a no-brainer. We know about the benefits that come along with having insurance. So why don't we have insurance for research? Now, we mentioned having a couple of horror stories, so here's your first one. I've used a fake name, but I promise you it's a real story. So Jack Doe is a proteomics researcher at a hospital in Toronto, Canada. Him and his colleagues had been actively searching for a protein, which we'll call protein A. They thought this protein could be used to test whether someone had pancreatic cancer. Jack and his colleagues proceeded to purchase a protein detection kit, which they used for 2 years and spent $500,000 before they realized the antibody in the kit was recognizing a different cancer protein, protein B. That antibody that they had been using did not bind to protein A at all. Now, with de novo protein sequencing as his research insurance, this catastrophe could have very easily have been avoided, and I'll get into how that looks in a bit. So I've given you a brief introduction, and now I'm going to go into detail about an industry problem that having research insurance in the form of de novo protein sequencing can address. Finally, in the last bit of the presentation, I'll cover the nitty-gritty processes and technologies behind de novo protein sequencing. So from our industry experience, and we have quite a bit under our belt now, most researchers address their research problems reactively, as seen in this chart. Our goal is to help make this chart look very different in the future. Instead of putting out fires, we want to help you prevent them. The facts are that research can cost hundreds of thousands of dollars, can take years to complete, and by the end of the day, it may not even be reproducible. If we look at the big picture, avoiding problems instead of solving problems should be the industry standard, but we've seen that it's not, and we're trying to get you to the right side of the chart, pun intended, by proactively seeking to avoid sticky situations. So there are a few ways in which your research can be insured, but one of the simplest ways to complete this is de novo sequencing. Now, I've mentioned de novo protein sequencing as a form of research insurance a few times, but I want to make sure that you know what it means. So what is it? Well, it's technology that uses mass spectrometry paired with some fun algorithms to analyze peptide data and consequently determine the amino acid sequence. This sequence includes isobaric residues such as isoleucine, leucine, and observed post-translational modifications of the protein. For those of you who are familiar with peptide mapping or intact mass analysis, to obtain an amino acid sequence, de novo protein sequencing is the only method that does not require a previous sequence to go off of, and guarantees deciphering point mutations, which, as you may know, is very important in variable regions of proteins, such as the 6 CDRs and antibodies. So I'm going to go ahead and hit you with some scary but really important information. In a publication completed in 2015 by Michael Weller, data was compiled from a variety of publications about the reproducibility crisis. And it was determined that 6,000 commercial antibodies from 26 suppliers, more than 75% of these antibodies were non-specific or did not work at all. Of the research experiences wasted in the USA, 36%, which is 10 billion US dollars, is assigned to biological reagents. And only 44% of all antibodies mentioned in publications can be identified at all. After some years, most of the published antibodies are not even available anymore. And out of 5,000 antibodies examined by the Human Protein Atlas, over 50% could not be used in the anticipated application. And may I just note that these are the applications that commercial suppliers themselves are stating. So how can it be solved? Well, the sequence information of antibodies through de novo protein sequencing is a powerful way to avoid antibody losses and guarantees essentially indefinite availability of an antibody through recombinant engineering. You're able to keep your research safe in a long-term economic way while ensuring your antibody is never lost. Additionally, with the sequence, you can directly tie your antibody to an antibody ID, so you're always able to keep track of what it is and where it is. So here we see 3 types of problems working with antibodies that can contribute to research irreproducibility. The first I'll discuss is the cross-reactivity in antibodies. Antibodies are theoretically supposed to bind to a target protein or residue. However, that's not always the case, and it can bind to many. When selecting antibodies to develop therapeutics or IVD tests, it's really important to ensure your research, which can be accomplished in a couple of ways here. By testing for off-target binding using both positive and negative controls, or a different method would be to obtain the amino acid sequence of your antibody, which could then be used to analyze the binding sites in silico. The second case we see is the wrong application of antibodies. As antibodies can be finicky, the varying experimental conditions can impact research results. If protein folding and binding vary, you could end up going around in circles, trying to figure out what's going wrong. Suppliers sometimes do have recommended applications, but that's not always the case. And there could still be differences even when the recommended solutions are followed. If you are proactive or reactive, having the amino acid sequence of your antibody of interest aids you in predicting the types of post-translational modifications that could occur or how different buffers could impact the binding affinity of your antibody. The only difference is, if you're proactive, you probably saved quite a bit of time and money. Now, with antibody variability, separate batches of antibodies can have variations, which would result in different outcomes in your research. For example, if you used antibody X back in 2018 and buy antibody X again now from the same supplier, you may notice inconsistent data from your research. Variability is the one that is the most difficult cases to control, as generally in industry, manufacturers themselves are most of the time unaware of the variability within their antibodies. Antibodies are characterized based on what they bind to, not their physical properties. So the variability observed could be for a variety of reasons. For example, genetic drift of hybridomas, or hybridomas produced from a new set of animals. With de novo protein sequencing, both the supplier and the researcher can obtain research insurance that addresses variability through protein sequencing. Suppliers could use an amino acid sequence to test batches of their produced antibodies. If there are any weird post-translational modifications, an amino acid sequence would help to determine small variations that were potentially missed from standard reagent protocols. In fact, one of our most proactive clients sent us a few samples at a time of different batches of their therapeutic antibody to ensure consistency of their product. Manufacturing high-risk drugs means that reactively looking for a solution could equate to detrimental effects having already occurred, which we know is far from ideal. Now, on the other end of this, researchers can proactively address variability by obtaining the amino acid sequence of their protein to ensure they consistently express the correct protein forever, and have absolute certainty of what antibody works best with their research. Now, another way antibody variability contributes to the reproducibility crisis in research is that monoclonal antibodies are generally thought of as monospecific. But this is not always the case. In a study done by Bradbury and al. In 2018, genetic diversity was reported in antibody heavy and light chains genes within an individual hybridoma. Now, if we take a look at this picture, we can see a variety of genes in the hybridoma and their reflected diverse alleles in red and green light chains in section A, as well as the black and blue heavy chains in section B. Clearly, there is much more than one potential antibody being created from these hybridomas. In this scenario, with different heavy and light chains, there are more than 9 potential IgG species that could be the one that they wanted. Out of 185 hybridomas that were analyzed, it was found that over 30% contained one or more additional productive heavy or light chain. So why do we care? Well, this means that the larger diversity of unexpected monoclonal antibodies could very easily be leading you astray, introducing multi-site binding or strange post-translational modifications, which could be why you're not getting consistent results. In fact, it was found that the most abundant mRNA transcripts found in a hybridoma cell line did not necessarily encode the antibody chains providing the correct specificity. It's clear that these chains can have a huge impact on binding affinity, efficacy, and bringing it back to the reproducibility of your research. When creating therapeutics or diagnostics, consistency is essential, and batch variations, as we know, could have harmful effects on patients. Now, some of you may be asking at this point, why not just use DNA sequencing in this scenario? Well, although DNA sequencing is a very useful technology, additional chains and post-translational modifications can be really difficult to capture compared to actually working with the antibody to determine the sequence. Now, we're all human, and if 2020 has taught us anything, it's that life can be unpredictable. So accidents way out of our control can happen. Now, imagine you're a researcher named Joe. Joe has worked in his lab for over 10 years and has had a huge impact in developing his team's go-to antibody. It's been used in countless research projects worth hundreds of thousands of dollars, and his hybridoma cell line is preserved in their freezer that the whole lab has access to and uses quite frequently. One dark night in October, a big storm hits and knocks out the power in Joe's neighborhood. Unfortunately, Joe's lab is located just 10 minutes away, and its power has also gone down. But, Joe doesn't think twice because his lab invested in a generator years ago for this very purpose. But however little did he know, the generator was very old and it never turned on. So his most prized antibody cell line has now died. So this may seem like a fictional horror story to you, but the unfortunate reality is that it's the reality for a surprising number of people. Maybe it wasn't a storm that cut the power line. Maybe the freezer was old and died all by itself. Or maybe someone dropped the cell line before it could even get to the freezer. The potential for accidents to happen is much larger than any of us would like it to be. We know of researchers that have spent hundreds of thousands of dollars trying to develop new protocols for their research and wasted months of their time trying to resurrect lost cell lines. It's unfortunate because if we had known about the problem at the time, or if they had known about us, they would have very easily have been able to resurrect their lost cell line. With de novo sequencing, providing the company with an amino acid sequence would have enabled them to recombinantly engineer their antibody of interest. And even if it's not a lab accident, say your supplier stops selling your go-to reagent antibody, it could take you a lot of time and money to test for a new antibody, which could negatively impact your ability to be successful in your research target. With the amino acid sequence, you're covered and you're stress-free. So with that story, I think I've covered my basis for providing you with a few user cases and a couple of horror stories. So I hope at this point you're starting to think of how to ensure your research. Now I'm going to take a bit of time to go into detail about how our lab actually completes de novo protein sequencing as well, or as we call it, our REMAP service, to provide you with your amino acid sequence. Our expertise lies in de novo monoclonal antibody sequencing, so I'll be tailoring the specifics to monoclonal antibodies. Here we can see a general 6-step overview, which I'll be going into more detail with. So when we receive our samples in our lab, our first step is to run a reducing STS page gel to determine the purity of the sample. In a perfect world, we would see 2 bands portraying the heavy and the light chains of the antibody, as seen in the middle column. But oftentimes what we see looks more like the column to the right, unfortunately. The blur that you're seeing is a result of contaminants, the like of BSA proteins, or even unexpected BSA antibodies. Now, in our early days, the column on the right would have presented a difficult challenge for us to overcome. But after completing over 2,000 sequencing projects, we have developed technology that helps us to purify samples like these, minimizing the extra contaminants. When we run digestions and put the sample through our mass spec, it's really important to limit any extra noise that could impact our ability to determine the amino acid sequence. So after the antibody has been purified, we separate out the heavy and light chains, as well as any glycans that are attached to the antibody. We then proceed to cleave the chains with a variety of peptides, which we like to call our peptide cocktail. The number of peptide used to digest the chains varies between 5 to 10, and the type of peptides are specifically chosen to cut the antibody at different amino acid positions. The diversity of these cuts allows us to optimize the peptide coverage of the antibody and really guarantee the accuracy of the data we obtain. Once the digestions are complete, we run the peptide through our thermo-Orbitrap Lumos or fusion mass spectrometers. The data we obtain from our mass specs is then converted to peptide sequences, and our REMAP proprietary algorithm is used to assemble these sequences into a draft amino acid sequence from N-terminus to C-terminus. Now, before we provide you with the final sequence, the draft sequence is actually reviewed by our bioinformatics specialists, who take the time to look at every single amino acid in the sequence to ensure absolute confidence in the sequence that we're delivering to you. Now, what you see to the right is the peptide coverage of the determined amino acid sequence. Generally, a 10-fold coverage, the confidence of the sequence is in the 90s, and it's still considered to be quite good. But the quality standard that we've set for ourselves is 30-fold coverage, which provides the highest industry standard of 99% confidence. Now, I'm going to make a special call out to our WILDS technology for providing an extra layer of research insurance in protein sequencing. When using a mass spec to determine a protein sequence, we rely on the differentiation of molecular masses of our amino acids to help us determine what amino acid we're dealing with. The difficulty with isoleucine and leucine is that they're isobaric amino acids, meaning they could have identical masses and they're not easy to differentiate. Within the CDRs of antibodies, there can be between 4 to 5 isoleucines and leucines, which means that if you're unable to differentiate between the two, then it could result in 25 different variations of your antibody. Now, if we're offering security of an amino acid sequence to ensure your research, 25 potential variations is not acceptable. So we developed WILD, standing for W-ion isoleucine-leucine differentiation. The W-ion is what actually allows us to differentiate between the two amino acids. When our peptides are run through the mass spec, they break up along the peptide backbone, as well as the constituent R groups, and lose a mass, creating the W-ion. This really enables us to determine which amino acid we're dealing with at a specific site, and ultimately provide you with an extremely accurate amino acid sequence. Through our industry experience and fine-tuning of our technologies, we're confident in our ability to provide de novo protein sequencing that we know will help you ensure your research, safeguarding it well into the future. So to conclude, having research insurance can aid you in saving a tremendous amount of time, sometimes even years, like my horror stories. It can also save you hundreds of thousands of dollars in wasted human and scientific resources that could be better spent accelerating your research rather than trying to get it back. And lastly, having research insurance aids you to stay on track and achieve reproducible, consistent results. And even if you don't think de novo protein sequencing best applies to your research as a means of insurance, I hope I've made you at least think a bit more about protecting what you worked so hard for and ensuring that whatever unpredictable events get thrown your way, your research will remain unaffected. So thank you so much for listening, everyone. If you believe your research could benefit from having insurance, and hopefully you do at this point, give me a call or send me a message. I would be happy to chat about different insurance applications and learn more about your research. My information is on the slide, and I'll open it up to questions.
Thank you, Monique, for a very good webinar this morning. I do see we have a couple of questions that came in, so I'm just going to go through them now.
Okay.
The first question is, can you handle antibody variations such as IgM?
Yeah, absolutely. So I know what I talked about primarily was IgG antibodies. But our mass spec and our technology enables us to actually handle all sorts of antibodies, including IgM. We've also dealt with nanobodies, and the species of the antibody doesn't have an impact on our ability to provide you with a confident amino acid sequence.
That's great. We have another question. It says, I've never done de novo sequencing before, so how do you know that the sequence is correct?
Right. So throughout our processes, we've implemented a lot of quality assurance factors, like for example, our bioinformatics specialists, triple checking it. But what really helps us too is our lab is in-house. So if for any reason we've completed the draft sequence and we're not comfortable or confident in what we're providing you with, we can very easily go back, run a few more tests, and make sure that what we're providing you with is as perfect as it possibly can be. And we're very confident in the sequence we provide you with, but if by any small chance, the sequence that you get isn't really working out for you, we are more than happy to go back in and troubleshoot the problem for you.
That's really great to hear. And we just have, I'm just going to ask one last question. Okay. Why not use DNA sequencing as this insurance policy?
Yeah, so DNA sequencing can often be a great method of insurance, but for really completely getting accurate results and ensuring that what you have is what you think it is, de novo protein sequencing is the best way to accurately get that information. Primarily just because with the amino acid sequence, you can really call out any post-translational modifications. And as I mentioned previously, oftentimes hybridomas have extra heavy light chains. And if you're going through DNA sequencing there, you might not pick up the different variations that may be within your hybridoma, or even if you do land on a sequence, it may not be the most high affinity binding one that that hybridoma is producing. With protein sequencing, we address all of that and really enable you to be sure about the research that you have in your hands.
Super. All right, well, I think that's all the time we have for today. So I'll let Monique wrap things up, and I'm sure she'll be happy to answer any more questions through email or a meeting outside this webinar.
Yeah, absolutely. Thanks so much, Jen. And thank you everyone for attending. And feel free to reach out to me at any time.